Good afternoon. We'll dedicate some time now to the visual system, which is a very important system, and both from an application point of view and from the research point of view. It has served as a model to study many other systems within the central nervous system, and to give you an idea of where the field is at this point. If you go to a meeting of the Society for Neuroscience that is attended by 35,000 people, maybe 20,000 presentations, a good 1/3 of the presentations have something to do one way or another with the visual system. To start with, I want to tell you that there are over 40 million people that are blind worldwide. And from those 40 million, over 1 million people are in the US. Now, why they are blind? Various reasons. The major cause of blindness is actually a relatively benign condition that can be treated, modified. That's cataracts, which is opacity of the lens in the eye, intraocular, and that see blurred and eventually you don't see. Fortunately, this is correctable surgically, and this is, of course, the usual procedures in developed countries, in this country, in Western Europe, in other developed countries, but in certain parts of the world, it's still a problem. Another cause of blindness is glaucoma, which is intraocular high pressure that results in atrophy of the optic nerve. Again, this can be treated in various ways and takes care of maybe 1/6 of the cases. Then you have a condition which is very important, actually the major cause of blindness in people that are over 65 years of age. That's what is called the age-related macular degeneration that affects about 10% of the cases of blindness. Next, you have various metabolic diseases, particularly diabetes, that results in retinopathies, inflammation and exudates in the retina and consequent eventual blindness. Finally, there is also trauma, which is not very frequent, but is there, a good 3% of the cases. And as you add this up, still you remain with a good 20-25% of the cases that are not due to all of these causes, and that put together many other causes of blindness. Now, something that to call attention at this point is the fact that all of these processes that are taking care of a good 75% of all blindness, all right, have to do with problems in the eye globe, more than in the central nervous system. So we can go now to sharpen a little bit the objectives of this presentation at this point. We have to discuss essentially the receptor apparatus, and then the control of pupillary size and of refractive power of the lens, and the effects of lesions on that. Then we'll discuss the concept of visual field, which is very important to have it clear, which is where you are going to locate problems in vision within the whole environment that you have visually access to. Finally, we'll discuss how the visual process starts at the retinal level and how this information is transformed in various retinal networks that are several complex structures, as we're going to see, and all of this to end up with the information being delivered to the brain in separate channels that take care of special attributes of the stimulus. That means that the stimulus is like deconstructed and the information sent in parallel to the brain that is going to put it together. We start as a sensory system, and we'll start with the stimulus. And the stimulus is always a form of energy. And in this case, it is electromagnetic radiation within a relatively narrow range of wavelengths, let's say roughly between 350 and 700 nanometers. If the radiation is, the wavelength is below 350, you are in the ultraviolet range and you don't see anything. If it's above the 700 nanometers, you are in the infrared range and you don't see anything again. Now we go to the receptor organ, which is the eye globe, right? And the eye works like an old photographic camera with some advanced features, such as automatic brightness control that is effected through pupillary changes in pupillary size, an automatic focus control that depends on the adjustable intraocular lens that we have. And then you have two classes of film in that camera. One film is fast, but this fast means that it reacts with very little light, but it's coarse grain and it's only black and white, and that's carried by what is called the rod system. It's one kind of photoreceptor. And the other, it will be just the reverse, is a slow film-like, but it's very fine grain and sees color, and that will be the cone system. And we are going to discuss that in more detail later on. Let's start now with the automatic brightness control that is carried out through the changes in pupillary size. Now, the pupil can react to light by becoming smaller, or it can react to darkness, in which it becomes larger. And these two processes are active processes. So you have these two independent processes, the pupillary constriction, that is due to contraction of an sphincter muscle, circular muscle around the opening of the pupil, that when the muscle contracts, the pupil becomes smaller. And this is under parasympathetic control. And on the other hand, you have pupillary dilation that is carried out by muscle fibers that are radial, in radial arrangement in the iris, and when they contract the opening, the pupil becomes larger. They are under sympathetic control. Now suppose you make a light flash to one eye only. What's going to happen? The eye that is exposed to light will show a constriction of the pupil. That's what is called the direct response. But if you take a look at the other eye that you have shielded from the light, you will see that it also constricts. That's what is called the consensual response. In here, you can make all kinds of combinations of problems in pathology, because if you cover the left eye and you flash a light on the right eye, and you see that this right eye constricts, and you take a look at the other one and it didn't constrict, what is the problem? Well, obviously, if the first one constricts, means that there is an input and an output that makes it that way. The input goes to the other side also, but what is failing here in the other side is the motor part. So you have a third nerve problem that provides the parasympathetic innervation on the left side. Or another example, if you cover the left eye and you put the light on the right eye and the right eye does not constrict, and you take a look at the other one and it doesn't constrict either, means that there is no input. The problem will be in the right optic nerve. All right, this is easy, relatively easy to understand and follow. Now let's look at the mechanism of the pathways that this reflex is following. You have the midline, then you have a left eye and a right eye looking at the world, looking at it from the top. And you see that if you put a shield on the right eye and you shine a light on the left eye, what's going to happen is that the left eye, the left pupil will constrict. That's the direct response. But the right pupil will also constrict, which is the consensual response. That's what we discussed before. Now, the outputs from the eyes, as we're going to see, will go through the optic nerve, all right? And the optic nerve, part of the fibers stay on the same side, and part of the fibers cross to the opposite side. But this kind of fiber that carry that information for the pupillary light reflex are going to terminate in networks of the area that is called the pretectum, that is in front or rostral to the tectum or superior colliculi. They have nothing to do with the colliculi themselves. It's just in front. But you can see that there is a bilateral input from one eye into both pretectal regions. Now, whatever happened to that, network that receives other inputs, including from the cerebral cortex, the output will project to the nuclei of Edinger-Westphal, as you can see, of both sides. And this output of the pretectum, as you see it here, all right, is to the ipsilateral Edinger-Westphal, and also some fibers cross the midline again and go to the opposite Edinger-Westphal. So at the Edinger-Westphal nucleus are these motor neurons that are preganglionic parasympathetic fibers, that they take this route of the oculomotor nerve and they go into the orbit and they terminate there in the ciliary ganglion, which is a parasympathetic autonomic ganglion, right, which contains a network with different other kinds of neurons, and that eventually will have an output that will take the route of the ciliary nerves to reach the sphincter muscle of the pupil. But as you can see, since the inputs for all this system goes to both sides, the other side will also carry on with the oculomotor nerve and ciliary ganglion and ciliary nerves, and the other pupil will constrict consensually. All right, to make it complete, you can put the fibers now from the right eye that will cross, go to the pretectum, and from the pretectum to the Edinger-Westphal nucleus of the other side. And you can see that these are these crossings, all right? And this crossing here is the optic chiasm, and this crossing here is the posterior commissure that we have seen in the oculomotor lecture, that the section of this posterior commissure will produce the pretectal syndrome, which is the paralysis of upward gaze, the pupillary disturbances that are occurring because of this mechanism, together with the eyelid retraction. All right, now let's go to the pupillary dilation to darkness, which is a sympathetic mechanism. Here you have again the middle line, and you have the left eye and the right eye. And now the fibers that are taking care of this reflex of dilation to darkness are mostly the cross fibers of the optic nerve, right? They cross in the optic chiasm, as we can see, we'll see later on. And just on top of the optic chiasm, there is a very tiny nucleus that is part of the hypothalamus that is called the suprachiasmatic nucleus. Very important structure. It contains actually the biological clock, internal clock that we have that has this periodicity of 24 to 25 hours. We'll discuss it in detail in hypothalamus. Now, hypothalamus is a small structure but has various networks within it. And what happens here is that part of the output of the suprachiasmatic nucleus goes or connects or projects to a portion of the hypothalamus that is called the lateral hypothalamus. All right, whatever happened there, then there are pathways that descend from the lateral hypothalamus all the way through the brain stem, which is midbrain, pons, medulla. They go into spinal cord and they come down to thoracic, upper thoracic levels, where they terminate in the gray matter of the lateral horn at this upper thoracic level. Has the name of ciliospinal nucleus, doesn't matter. All right? And that is on both sides. Now, this nucleus in the spinal cord contains preganglionic sympathetic fibers that take the route of the paravertebral chain and climbs that chain by passing the ganglia of the lower cervical upper thoracic region, and they terminate in the uppermost ganglion of this chain that is called the superior cervical ganglion. Again, it's an autonomic ganglion, contains a network, things happen there, and the output of the superior cervical ganglion is taking the route of the carotid, the pericarotid plexi of nerve fibers, and with the ophthalmic artery goes into the eye, and and eventually reaches the iris where it innervates these radial fibers and the pupils dilate. All right. Now, one important syndrome that affects these pathways, the sympathetic pathway to the pupil, is what is called the Horner's syndrome. All right. In the Horner syndrome, you have the system as outlined before. Let's put it together. It's the same as before, all right? But these pathways, these sympathetic pathways here, are at failure in the Horner syndrome. So what is the patient going to show? Well, to show a small pupil, let's take a look at it. There. On that side, it's going to show a small pupil with regard to the opposite side. Also, it showed a partial drooping of the eyelid, not the ptosis that you see in the third nerve palsy, in which the eye is completely closed, but this is just a narrowing of the palpebral fissure. So when you see a patient that has that pupil smaller than the opposite and a pseudoptosis, this lowering of the eyelid, and has also other phenomena such as vasodilation in the face and lack of sweating, you say, well, this is a Horner syndrome. The sympathetic on that side is a failure. So what can be the cause of that failure? Well, there are vulnerable sites within this general mechanism, all right? And one of these vulnerable sites are the paravertebral chain. So the paravertebral chain is a series of ganglia and nerve trunks that interconnect them that are just very close to the midline, very close to the spinal column. And it so happened that if you have a lung cancer, particularly of the apex of the lung, so this is an apical cancer, when it expands, you have right there, you have the paravertebral chain that is affected. So the patient may show a Horner syndrome, sometimes even as a first sign of the lung cancer. So you have to keep it very clearly in mind. There is another vulnerable site that is the brainstem tegmentum, where all these descending pathways are going through. Well, we have seen that in the most common syndrome that you see attributable to the brainstem, that you may remember is the lateral medullary syndrome or Wallenberg syndrome, in which you have, it's a cross syndrome because it's brainstem, so you have pain and temperature loss on the same side of the face and the contralateral side of the body, and in addition you can have a Horner. Why? Because in that tegmentum of the brainstem there are also these descending pathways that are coming from the lateral hypothalamus, and they can be caught there, for instance, with the same pathology of an infarction in the pica territory, and give you the Horner syndrome, in addition to the other components of the cross syndrome. All right, so we finished with the pupils. Now we should go to the automatic focus control that is called, the phenomenon is called accommodation. All right? So accommodation is depending on the lens system that can be more or less adjusted, as we're going to see in a moment. Here you have an important consideration here is to understand that we are dealing now with the refraction power of lenses. All right? What is the refraction power? It's how much convergence the light rays entering through one side of the lens are going to converge on the opposite side. These are positive lenses. All right, the focal length, that depends on the focal length of the lens, right? Because if you have a lens that is just 1 centimeter in radius, which is the focal length, let's say, it will be a very curved lens to have just only 1 centimeter in focal length. But instead of 1, you have 50 centimeters, you see that the lens will be much shallower, less curved. Now, focal lengths are values that are not non-additive. So the ophthalmologist, the eye doctor, is not good, that wants to try several lenses to see which one is better for you or whatever, is not going to use focal length. He's going to adopt a system that is called the diopter system. And you have to have clear that 1 diopter is the reciprocal of the focal length in meters of that particular lens. All right? So here you have, for instance, an eye globe, all right? You have here the cornea, and here this is the whole globe, this is the cornea, and this is the intraocular lens. All right, so let's say the cornea has 43 diopters, and it's fixed. You know, the cornea never changes. 43 diopters. It's a very really converging kind of lens. On the other hand, the intraocular lens, all right, when it's fully relaxed, what do you mean fully relaxed? When you're looking at the distance, you're not doing any effort to see something close or anything. You look at the distance. So the lens is fully relaxed. Still has 15 diopters. Right? So you have a total in the lens system that, you know, the inside of the lens, the vitreous humor that is here contributes to some extent. But the main thing is these surfaces. The cornea, 43 diopters, the intraocular lens relaxed 15 diopters. So you have, in that way, in that particular case, you have that parallel rays that go through this lens system are going to focus exactly on the surface of the retina, and you see perfectly well. Right? Now, on the other hand, when you see something close to you, you need a more power in the lens, more refracting power, to bend the rays in such a way that it will still fall sharply on the retina. And for that, you have your cornea of 43 diopters, always the same, but your lens will change, all right? And can increase its power initially from 15 diopters. It can go, particularly in babies, up to 29 diopters. So it's, when it's fully accommodated, you cannot do it more than that, right? So have this in mind, and in this case, of course, now you have a very close object. You don't have parallel light rays from the distance. You have a close object like that is going through the lens system, and through the increased power of the lens, intraocular lens, still will project sharply on the retina. All right, what can happen? By the way, this accommodation, all right, is part of the near vision reflex that we discussed in eye movements when the eyes converge. Remember we said that the eyes converge, the accommodation occurs, increasing the power of the intraocular lens, and the pupil constricts. All these three phenomena occur at the same time when you want to look at something close to you. Now, how come that this lens can become more powerful? Well, what happens is that the lens, right, is supported by a ligament, a round ligament all around, right? And this round ligament is connected to a sphincter muscle again, which is the ciliary muscle. When that ciliary muscle constricts, look what happens. The opening of the muscle becomes smaller. When it becomes smaller, the ligament that holds the lens relaxes, and the lens is allowed to become more convex. That's the whole process of accommodation. All right, problems with accommodation, you have refraction errors, for instance. You have, when the eye is normal, it's called emmetropic. And in this case, you have an eye globe, and you have your parallel rays that are converging exactly on the retina. But you have cases, many cases actually, in which the AP diameter of the eye is longer than normal. These are people with large eyes that you see. And these people, look what happens, that if you have now the parallel rays coming into the eye, they converge according to the power of the cornea and the crystalline lens, but the focus occurs in front of the retina. Means that at the retina itself there is a blur. Right? That's a myopic eye. All right? And the way of correcting a myopic eye is putting a lens, in your spectacles, you put a lens here that is negative. It's just the reverse of this one, that will actually decrease the power of convergence and the eye doctor will fit that lens so precisely that now the focusing will be exactly on the retina and you see clear again. You have also the reverse case that is called the hyperopic eye, in which the AP diameter of the eye globe is shorter than normal. In this case, you can see that the light rays are going to focus behind the retina. So you need to increase the power of convergence, so you put a positive lens for that purpose. That's something that occurs, this deficit in refraction occurs also with age when the accommodation function that makes the lens variable according to needs fails. And so at certain age, the lens doesn't converge enough the rays, and you need an additional lens to read, for instance, because when you read, it's at closer distance. So you need, again, in this case, a positive lens that will make the whole system more refractive and will get your focus on the retina itself. There is a third problem in refraction that is now due to problems in the cornea, because this cornea is really a segment of a sphere, right? But if that segment of a sphere is not regular, becomes like distorted, the sphere, is not a sphere anymore, then you have what is called astigmatism. And in these astigmatic eyes, the eye doctor will figure out the axis where the astigmatism occurs and will fit a lens with just a cylinder taking care of that particular axis. All right, that's about that in accommodation. You should know that the mechanisms for accommodation are very similar to those that produce pupillary constriction, because after all, they are both part of the near sight system, right? Pupillary constriction and accommodation. Well, there are cases in which there is a dissociation between the two. And in the old times, where tertiary syphilis was in fashion, what was happening is that these patients had relatively small pupils and pupils that did not react to light with further constriction, but did constrict further on accommodation, on near vision. So that's what is called the Argyll-Robertson pupil. That was a classic, and maybe still is a problem of a question or something of that sort. You don't see much of it at this point. All right, let's go now to something more that has to do with the bedside examination. And that's, we have to have a concept of what's called visual angle, then to apply it to the size of visual fields. What's the visual angle of an object? When you look at an object, all right, you see a given size, all right? But that size, it reflects actually the visual angle subtended by the object. For instance, if you have the eye globe here, and you have now an object, let's say that is 1 inch in length, and is placed at 5 feet from the eye, you have now the conditions that this object is subtending a certain angle. And it so happened that if the object is 1 inch at 5 feet, the angle is 1 degree. What does it mean? That 1 degree also is an object that is 2 inches in length at 10 feet. So these two objects have the same visual size, 1 inch at 5 feet or 2 inches at 10 feet. That's the concept that the objects of regard, their size is measured in degrees. Now, another example, for instance, you have the same situation, right? 1 inch at 5 feet is 1 degree. We know that. But what about the same 1 inch at 10 feet? Well, you can see now that the visual angle is much smaller, and is in fact half a degree. So this is a concept that you should definitely have clear, because problems in the visual field, as we are going to see, are referred as locations in visual angle within that visual field. Let's talk about visual field. What's a visual field? When each eye has its own visual field. Of course, when you have both eyes open, it will be a binocular field, which will be an overlapping of the fields for the right and the left eye. That's an obvious thing. But let's say we talk about one eye at a time. Let's say the right eye. What is the visual field of the right eye? Well, if I look at it straight ahead, everything that you see without moving the eye is the visual field of that eye. All right? It has a size, all right, that you can more or less chart by asking the patient to report when he sees your finger or when he sees something moving, actually, and you can plot it, right? And when you do that, you see that the extent of the visual field depends majorly on the stimulus. And the stimulus that gives you the maximal extent of visual field is movement, detection of movement. So you will see something moving, but you won't know what it is until it comes closer to the center, right? So let's say for the left eye, when you plot the visual fields, it's like you're looking at it. So you're looking at this one with your left eye, and your left visual field is something of this sort, and the right visual field is like that. So it has a peculiar shape of different extents, all right? It's very much extended. Here in the visual field, as you can see, this is the center of fixation, but you're looking straight ahead, and you have quadrants, right? So you have, that I'll tell you the quadrants in a moment. Apparently, it's first the extent of the field. You see, this is 60 degrees. This point is 60 degrees. And this point is 70 degrees. And this point is 100 degrees. And towards the nose is 60 degrees. All right? And then you have a cut here that is given usually by the nose that doesn't allow you to see if you put the finger behind the nose, right? So that cuts off at 45 degrees. All right, so as I said before, you can divide it into quadrants, all right? And from that standpoint, you have here all the quadrants that are outside of the center of fixation. They are called temporal quadrants because they refer to your temple, right? Whereas the next ones, these ones, are called the nasal quadrants because they go towards the nose. Now, all of these are in space, are not inside you. The visual field is in space, right? And you have temporal quadrants outside there and nasal quadrants towards the nose. Also, you can see that the quadrants can be superior quadrants or can be inferior quadrants. So essentially, you have a superior temporal quadrant, an inferior nasal quadrant, and so forth and so on. Now, when you keep and you examine one eye at a time, but when you are, normally you keep both eyes open, so you have an overlap of these two visual fields into what is called a binocular field. The binocular field looks like that, right? It's one overlapping the other. All right? And you can see here that there is a center portion of this binocular field, all these, all right? All these that is seen by the two eyes. But then you have, on the temporal side of the visual fields, you have what are called the temporal crescents that are seen just monocularly, with one eye only. All right, so these are the visual fields. Now you can have, in patients, you can have visual field defects, which means that part of the fields don't see so well, right? And you have one example here in which there is an area of blindness in one eye only. This is what is called an anopia or a scotoma. It is a scotoma, right? Scotoma. Then you can have another patient, or even the same patient, that the other side of the the visual field of the other eye is totally blind, right? And that's called an anopia. So a scotoma is a small part of the visual field that is blind, and anopia is the whole eye that is blind. Right, but you have other things too. A pattern like this, that you have that the left half of the left eye and the left half of the right eye are with problems, all right? So see, this is half of the field is called the hemianopia. And because it's on the same side for both eyes, it's called homonymous. So this is a homonymous hemianopia. You have another case in which you have, again, the left side of the left eye, but then you have the right side of the right eye. It also has fields. It's also a hemianopia, but this is heteronymous, and is also called bitemporal because of the temporal fields that are at deficit. Right? Finally, you can have cases in which only the temporal upper quadrant of the left eye is involved, and in the other eye is also the left temporal, the left nasal quadrant involved. All right, they are also now there are partial problems in vision, but they don't take half a field; they take only a quarter of the field. So instead of calling it hemianopia, you call it quadrantanopia. And since it's on the same side for both eyes, this will be a quadrantanopia homonymous. All right, these are examples of field defects. Now we have to go into the nitty-gritty story, which is, at this moment, the retina. The retina is a phenomenal structure. It's very laminated. You see? There is one layer here with this pigment, and then there is the receptor layer, and then the cell bodies of the receptor layers and all the other kinds, and then a synaptic layer, and then again a cell body layer and another synaptic layer, and finally the ganglion cells that project their axon to the brain. So there is a lot of processing that occurs in the retina, and that's what we have to go through in a little more detail. But I'm going to do it cartoon-wise, and I'm going to take just one element of each of these layers, just to try to make it somehow didactic. So now I'm going to show you a retina reduced to the bare minimum. So we have this layer of pigment epithelium. This is the single layer layer in the single cell layer in the retina. All right, we have a flattened cell body and has like expansions or protrusions towards the next layer. Right, this is the nucleus of the cell. Now this pigment epithelium has like shields, so goes around the next cell type, which is the receptor cell. And the receptor cell now is going to connect with another cell, which is the bipolar cell. And the bipolar cell, in terms, is going to connect to the ganglion cell. And the ganglion cell is a real neuron, all right? And the axon of this neuron goes to the brain. Well, that's the bare minimum. But you see that these collateral branches here connect to two other kinds of neurons. One is a so-called horizontal cell that goes back to the receptors, and the other is the amacrine cell that goes into the ganglion cell layer and various connections with the ganglion cell and with the terminals of the bipolar. So this is essentially a retina reduced to the bare minimum. Take into consideration, it looks like an online kind of transmission of information from the receptor cell to the bipolar, from the bipolar to the ganglion cell out. And then some lateral processes that occur, mostly inhibitory, that are carried by horizontal cells and by amacrine cells. All right. Now, the retina has imperfections. The one that created it really didn't know much about optics, and it's very imperfect. I'm going to show you in a moment. For instance, you have here the same cartoon that I showed you just now, all right? But now it's added some information that here at the pigment epithelium layer is the sclera, which is the tough covering of the eye globe, whereas at the level of the ganglion cells is the vitreous humor that fills the eye globe. What does it mean? It means that light that enters the eye, all right, has to go through all the layers in order to get to the receptors. So when light goes through tissue, it gets distorted somehow, right? Because the refraction power of the tissue, the transparency, and so forth and so on. So this is an imperfection, that the light has to go through tissue in order to get to the receptors. How nature compensated by that? Well, this thing makes it an inverted retina. You see, it's like it doesn't follow intuition at all, right? How is it corrected? It's corrected by the formation of the fovea, and the fovea forms in this way. You see, this is the layer of pigment epithelium, these cells up there, all right? And now, from the receptors on, all the other chain of cells in the center portion, they get like bend. See? Another one that bends, and another one that bends. And the end result of this bending, we are in the center of the retina here. The end result is the formation of a pit. And that pit is called the fovea. And the dimension of the fovea is something like 5 degrees altogether. It's quite small, all right? And at the very center of the fovea, you can see that the light can hit directly the receptor here. And that's what is called the foveola, that is about 1 degree or something like that. This is the gadget to compensate for the fact that light has to traverse all the layers in order to get to the receptors. And you can see that the more you go towards the foveola, the less tissue the light has to traverse, and therefore the less distortion. Now, there is another imperfection of the retina, and that is the fact that has quite a chromatic aberration of the lens system. So if you remember your college physics, optics, or whatever, chromatic aberration has to do with in a perfect lens system, there is no chromatic aberration. But you have other shapes, right, of glass, let's say, that are like prisms. And when light hits a prism on one side, the other side gets the spread of all the wavelengths, and you have all the colors of the spectrum. All right? So that's a prismatic effect that should not occur in ideal lenses. In the lenses of the eye globe, there is still some prismatic aberration, so chromatic aberration. What does it mean? That the light, white light that comes into the eye, right, gets a little bit decomposed into different colors, and therefore when it hits the retina, it again is a blur, because each wavelength is going to be refracted at a different power. So how the nature compensates that? Well, you have here a contracted version of the above, right? Now the fovea is here, that's the 5 degree, and you have the rest of the retina, right? What happens is that all the layers of the retina, except the receptor layers, all right, is pervaded by the presence of a yellow pigment. And it's like that, you see? All these layers contain a pigment. And that pigment extends for a total of about 20 degrees in the fixation part, and that's what is called the macula lutea, and colloquially is called the macula, right? And what happens here is that when the light goes through all these layers that have the pigment, the blues are essentially filtered. Several parts of the spectrum are filtered so that the distortion at the retinal focus is less marked. So that's the way that compensates for the chromatic aberration, the presence of pigment. The final aberration, the final imperfection is what is called the blind spot, all right? That you have done, I'm sure most of you, the experiment of having a spot here, and depending on where you put it, at some point the spot disappears, all right? And this represents the head of the optic nerve. Now, optic nerve are these axons from the ganglion cells. So when there is the head of the optic nerve where all this collects, doesn't have receptors, and therefore is blind. The head of the optic nerve is blind. And the resultant projection into the visual fields is a spot that the person doesn't see in that spot. And that's what is called the blind spot or physiologic scotoma. Right? So remember retinal imperfections. You have to keep it in mind. The retina is inverted, it's compensated by the fovea. The retina has the lenses have chromatic aberration, is compensated by yellow pigment in the macula, and finally the head of the optic nerve is blind. Now, you don't see blind spots, and you have one blind spot for each eye. It's the role of the brain to compensate for that. What is called, the brain feels the scotoma. Hi, everybody. I'm going to continue with the material that was interrupted this morning because of some technical difficulty. And we were at the level of the pigment epithelium, which is actually the first layer of the retina that you may remember that is the closest to the choroid and the farthest away from the entry of the luminous stimulus. So this pigment epithelium is a single layer of cells, flattened cells, these ones, all right, that have these protrusions that go between receptors, as we're going to see in a moment, and is against a vascular membrane, which is the choroid. Now, these cells contain a pigment, which is melanin, and that's why it's called the pigment epithelium. And one of the main functions is to provide for the active transport of various metabolites between the receptor cell and the blood, because the receptors themselves, the receptor cell and receptors, are not receiving blood supply from the central retinal artery, which pervades all the other layers of the retina but doesn't reach the receptor layer. In any case, this active transport then occurs both ways, from the receptors to the pigment epithelium and from the pigment epithelium to the blood supply. Now, in addition to that, the pigment epithelium has other functions which are very important. You see the receptors themselves, like here, are represented by this pile of discs. The discs are constantly shedding from the tip of the receptors. And one of the functions of the pigment epithelium is to remove those debris by phagocytosis. That's what is represented here. Then we can see what happened when there is light. When there is a shining light on the retina, what's going to happen is that, of course, these functions that we discussed before are still there, but you see that there is a migration of the pigment towards the processes, as if shielding receptors from each other. At the same time, as we're going to see in the chemistry, there is a byproduct of the retinal pigment itself, of the visual pigment, that a step that coincides with vitamin A. And that vitamin A now is picked up by the pigment epithelium that retains it as a repository during the dark adaptation period. Very good. So that's about that for the pigment epithelium. Remember, active transport, shielding of receptors through migration of the pigment, and phagocytosis of shedded discs at the tip of the receptors. Now let's go now to the receptors themselves. There are two types of receptors. One type, as you can see, there is a cell body and there is an initial cilium from the cell body, and then you have this pile of discs. This pile of discs are the result of pinching off the surface of invaginated portions of the membrane. You can see them here in this other kind of receptor, in which you see that the membrane has invaginated and has remained that way. That's typical of this kind of receptors without the cones, where these ones that are the rods, there is a pinching away from the initial membrane, and you have free-floating disc piles in the receptor themselves. But it's an important concept to have, because even when you see a free-floating disc in the cone, you have to know that what is inside the disc represents extracellular space, and intracellular space is what is between discs. I hope that I'm making this more or less clear. Now, these two types of receptors have various qualities that differentiate them. First of all, the sensitivity. You have that the rods have very high sensitivity, which means that you need very little light to activate them. Whereas the cones have very low sensitivity, you need a lot of light to stimulate them. Another feature is the acuity. The acuity means how close you can see two points, let's say, that you can still see it as 2 points. If they become closer, then you fuse and you see only one instead of two. That's the acuity value. All right. The rods have very poor acuity, whereas the cones are very high in acuity. Now, what about what it's called here the highest density? You will see that the rods predominate in what is called the peripheral retina, whereas the cones predominate in the central retina. Another feature is the visual pigment that these receptors contain. And this visual pigment is single in all the rods. All the rods have the same visual pigment, only one, all right? Whereas the cones, they can be segregated in at least 3 families of cones, one where the visual pigment is most sensitive to red, another to green, and a third one to blue. And you need the 3 of them to see color properly. Now, what kind of vision then they subserve? When if you see that the rods, all right, predominate in peripheral retina, and they are very sensitive to light but very poor acuity, and they don't see color really because they have only a single pigment, this is characteristic of night vision. All right, and night vision is also called scotopic vision. So scotopic of night vision. On the other hand, the cones that are just the reverse, the predominant in central retina, they are very low sensitivity but high acuity, and they have the 3 types of cones to resolve eventually in all kinds of combinations and the perception of color. This is typical of daylight vision that is also called photopic vision. So from all of this, you can already infer a very important concept, and that is that in the retina, there is 2 kinds really of retina. One that is central, which means around the center of fixation, and the other one that we discussed this in visual fields, right? That there was a very sensitive portion of the retina that had the fovea, the pit, so the light doesn't have to traverse much of tissue, and also there was a yellow pigment that was pervading most of the layers, acting as a color filter, and that central portion of the retina is where the cones predominate, and they deal with daylight, as opposed to the peripheral retina that deals with nightlight. I'll give you an example of a very simple observation. If you look at the starry sky, let's say not in New York City, but if you go a little outside and you see the sky with stars, all right, and you see that the stars are of various magnitudes, and if you look at them straight, you will not see, that means if you put the image of a particular star in the center of your retina and the star is very dim, is of 1/3, 1/4 of the magnitude, then you won't see it. But as soon as you look at it from the side and make that image come into peripheral retina, you immediately see it. All right? That's an example of what is scotopic vision. Really. All right. Now we have to go into the first step of the visual process that we haven't touched yet. First of all, let's say here is an example or a cartoon of a receptor, all right? And you have these free-floating discs, one on top of the other. And remember, and you can see that here because of the color Q, that the inside of the disc is like extracellular, it's like this, all right? Whereas what is bluish is all intracellular, which is between the discs, but not inside the discs. All right, so let's take a look at what happens when there is no light, no stimuli. You are in darkness, all right? And here I represent one of these discs, all right? And I even did it in an incomplete fashion here, which is not correct, but it's just to remind you that the inside of the disc is extracellular space. All right, in the membrane of the disc, there are a particular kind of protein molecule that traverses the membrane 7 times. All right, something like that. Well, if you remember from all your past learnings, this is typical of a so-called G protein coupled receptor. All right, and this protein here is acting like a receptor, right? And it's a protein that is called opsin. Now, embedded within the opsin is a chromophore group, as you can see here, that I just passed it through, all right? And this chromophore group is such that has in the chain, it has a bend in carbon 11. So this kind of chromophore group that is embedded within the opsin, within the protein, is called an 11 cis retinal. That's the name of that chromophore group. 11 because it bends at the 11 carbon, and cis retinal because that's the arrangement in space. All right. So this now, the opsin, which is the protein crossing the membranes, and the chromophore group, the 11 cis retinal, together they form what is called the visual pigment. All right. These cells has channels, all right, ion channels, and has an ion channel for sodium, for instance, ionotropic, all right. This channel is maintained opened by cyclic GMP. You see that, remember that we are with the light off, all right, and you have here a sodium channel that is open, maintained open by cyclic GMP. So there is, the first thing that happens is that sodium comes into the cell. If sodium comes into the cell gets depolarized, right? And now this is strange, right? Because you see that there is essentially no stimulus, there is no light. And however, the cell is depolarized by this lack of stimuli. It's an exception to the general rule in the entire nervous system. All right, another thing that you should know is that the membrane, within the membranes, there are, the G proteins, because this was a G protein coupled type of receptor, all right, the opsin. Now you have the G protein itself with an alpha, beta, and gamma subunits. Now, before I continue, you see, you recognize that this is G-coupled receptor molecule, all right? Actually, was the first G protein coupled receptor discovered, and it was called the G protein, the corresponding G protein was called transducing, because it transduced light into a virtual electrical happenings. That has been replaced today, and this protein is called the G sub T, reminding you of the transducing designation. Anyway, you have this molecule floating here. Another molecule that you have in the membrane is a phosphodiesterase, all right, with the two gamma subunits and alpha and beta. You don't have to remember all these details, but know their existence at this point, and there is no light. So there is no activation of anything except for the fact that they have the sodium channels permanently opened in darkness through the presence of cycle GMP. Now let's put some light. We shine a light. Light's on, all right? You have the same arrangement, all right? But what's happening is that the chromophore group that was bent at the 11 carbon, now the chain gets straightened. And so it's not anymore an 11 cis retinal, it is what is called a all-trans retinal. You know, retinal, as you may know, of course, is an aldehyde, right? It's retinal. It's an aldehyde all the time. But this is a new form now, in all-trans. And this is the first step of any visual process, is the action of photons on the all-trans retinal and transforming it into the of the 11 cis retinal that gets changed into all-trans retinal, right? Now this visual pigment now is activated, and it's called activated rhodopsin, as the name, right? So that molecule, that the opsin remains the same, but it's the chromophore embedded group that changes, and that makes it now an activated rhodopsin. All right. Now, what happens when that happens? When the carbon chain straightens, you see that the G protein that was floating there gets attached, gets coupled to the opsin, and when it's coupled, it dissociates its units, and it dissociates its units so that the alpha unit remains attached to the opsin, but the gamma and beta units now steal away the gamma subunits of the phosphodiesterase, and that makes the phosphodiesterase activated. It's an enzyme. It activates, and what does it do? It phosphorylates the cycle GMP, and that becomes just GMP without the cycling, and that process results in the closing of the potassium channel, of the sodium channel, meaning that the cell, by the action of light, becomes hyperpolarized, just the reverse as in any other part of the nervous system. All right. Any questions on that? All right, it's more or less self-evident. All right, now we continue. This kind of biochemical or photochemical mechanisms have electrochemical correlates, right? And it goes like that. If this is a rod, you know that the light is off at this point, right? And the sodium channel is open, the cell is depolarized and releases its transmitter. Transmitter release. That is usually, most probably is an excitatory amino acid of the type of the glutamate type. So we are in darkness, and you have this receptor cell releasing transmitter. All right. Now we go into light. Light on. What's going to happen? Well, your sodium channels now close because of the hydrolysis of cyclic GMP. The cell consequently becomes hyperpolarized, and the release of the transmitter is stopped, or I would say is decreased. In this case is increased, in this case is decreased, and is all counterintuitive because it's just the reverse as any other part of the nervous system. All right, these are electrochemical correlates. Right, so we just finished reviewing the structure and function of receptors. And at this point, we should go into the further processing of the information by retinal networks. That, as you remember, the retina is a quite complex structure with several layers and several neuronal components. The first thing is what we called initially a through pathway that will go, let's say, from receptors to bipolars, from bipolars to ganglion cells and out into the brain. Now, this first step of the receptors to bipolars are one of the different characteristics that this system has in relation to others. First of all, the receptor cell releases a transmitter in darkness, as you remember from before. That transmitter is probably glutamate, and as you can see, when it's released, it's going to excite a certain kind of bipolar cell, but at the same time is going to inhibit another type of bipolar cell. This is a clear example about that the action of a transmitter is actually determined by the receptor that grabs it. In this case, in darkness, the transmitter is released and is really activating, exciting a bipolar cell of the depolarizing type. All right, this bipolar cell discharges and in turn will activate a ganglion cell, and the ganglion cell discharges and sends the information to the brain. Now, this ganglion cell, as you can see, is discharges in darkness, and that's why it's called the ganglion cell of the off type. All right, at the same time, the transmitter is inhibiting the other type of bipolar cell that is called a hyperpolarizing bipolar cell. If you now introduce light instead of darkness, you see that the transmitter the transmitter release is decreased, and therefore the inhibition on the hyperpolarizing bipolar cell decreases. This cell, therefore, that is released from some inhibition is going to discharge, and the discharge is going to activate another type of ganglion cell that, as you can realize, is activated in light. And that's why it's called a ganglion cell of the on type. All right, in addition to this through processing, there are also lateral processes that are all of inhibitory nature, and that they are actually carried on by horizontal cells that carry inhibition in a lateral fashion, both to some bipolar cells and even to backwards into the receptors. And also by amacrine cells, of which there are numerous types, particularly groups that are characterized by the presence of particular neuropeptides that are differentially occurring in these groups of cells. In any case, these amacrine cells are making contact with both bipolar cell terminals and also ganglion cells. So in that way, in this lateral inhibitory fashion, this processing of information is such that it are going to determine the characteristics of a particular ganglion cell. Of course, neurophysiologists have revealed the phenomena that occur at each of these cell types. Each cell type has been blessed with intracellular recordings, and a lot is known about the function of each of these various cells. But that information we have to compress it at this point and reduce ourselves to the ganglion cells that will have characteristic, in addition to the off-on type, other characteristics that we are going to read in a moment, that are the result actually of the whole processing of information that is picked up by the receptors and then processed through all these networks that we mentioned before, and that results now in the characteristics of the ganglion cell that we'll review next. Now, the concept of receptive field. Now you have to understand it from the standpoint of an animal experiment, all right? You have an animal, let's say a monkey or a cat, whatever, is anesthetized with the eye open, one eye open, and then you have in front of the animal like a hemispheric field, like half a sphere that makes like a screen. And now you put an electrode in one ganglion cell of that retina. And now you start putting spots of light on that screen. And you will see that at some point, at some place in the screen, the neuron discharge will change. Either it will become more frequent, the discharge, or less frequent, the discharge. It can be one thing or the other. And so in these terms of this kind of receptive field, you can map it, right? Because you can put these spots in the hemisphere, and you end up with a piece of that screen, that anything that you put as stimulus within that piece of screen will make the discharge of the ganglion cell change. So that defines what is called the receptive field of that particular cell. And have cleared that the stimuli at this point are just spots of light. Right? From that standpoint, you can discover that there are two kinds of ganglion cells that react differently to these spots. For instance, if you have here the frequency of discharge of the neuron, all right, and you have here the size of the spot in degrees of visual angle as usually, you see that the particular ganglion cell has a certain frequency of discharge, all right, at this level. Now, if you put a stimulus that is only 5 degrees, a small spot, you will be a little bit of increase in the frequency of discharge. If you increase the size of the spot, it will increase even further. And if you keep on increasing the size of the spot up to 15 degrees in this case, the neuron discharges more and more frequent. All right? At some point, if you increase even further the size of the spot, you see now that there is a decrease in the frequency of response. And if you make it even larger, it will decrease even more. And if you make it even larger, nothing will happen. What does it mean nothing will happen? It means that it's outside of the receptive field already. It's so large the spot that overflows the receptive field. Now, what this can mean, this kind of graph? Well, it means that that receptive field of this particular neuron, goes out to about 25 degrees, as you can see, because it changes. But it changes differentially. At the beginning, it increases, and at some point starts decreasing. Increasing means that it's excited, but this decrease here means that some inhibitory influences are added in the so-called periphery of the field, right? Because it occurs here when the stimulus is beyond 15 degrees. Now, this kind of neuron that is characterized by this center portion that gives excitation and a peripheral portion that gives inhibition, that's what is called a center-on, surround-off ganglion cell. There is the opposite also. There are ganglion cells that are just the reverse, that they will be, as you can see here, in this graph, which is a mirror image of the other one, you will see that this kind of neuron, when the spot is small, gets inhibited, and when the spot begins to be larger, responds with excitation. So it's like the center, the receptive field in the center portion is off, and the peripheral portion is adding excitation, which means that is a surround on. And that's indeed what this neuron is. It's a ganglion cell that has a center off, surround on receptive field. Is that more or less clear? I hope so. All right, because this is a concept that sometimes is difficult to grasp, but I'll give it to you in some other way. So these ganglion cell receptive fields, that's what we are talking about, right? Again, I'm going to insist, these ganglion cells receptive fields are the result of an enormous amount of processing and transformation in the other layers of the retina. This is actually the neuron, the ganglion cell, is going to be an axon that delivers the information to the brain itself. All right. Now, the examples that I just gave you in the graph means that the organization of the receptive field is concentric, has a center-surround antagonistic organization. And from that standpoint, I showed you two types of ganglion cells. Here, for instance, this is the receptive field that is a center-on, surround-off type of ganglion cell. So any spot that is placed in this center portion will result in excitation of the neuron, increase the frequency of discharge, whereas any spot that is in the surround will decrease the firing rate of that neuron. So this is essentially what is shown here. Suppose that the resting discharge of that neuron is something of this sort, right? Now you put the stimulus in the center, you will see that the frequency of response increases. On the other hand, if you put a small stimulus in the surround, it will decrease the stimulus, the frequency of the response. So this is a typical center-on, surround-off ganglion cell. I'll give you the opposite, which I gave you in the other graph. Here you have a center-off, surround-on. That means that if you have here the resting discharge and you put a small spot in the center, it will decrease the frequency of response. If you put a little spot in the surround, it will increase the frequency of the response. So I hope that you have more or less clear the concept of these two types of ganglion cells that you are able to observe by using spots of light within the visual field and delineating the extent of the receptive field and also its characteristic center-surround antagonistic organization, which means something in the center excites the cell, something in the surround inhibits the cell, that's one type, or the reverse, that something in the center inhibits the cell and something in the surround excites the cell. All right. The problem is that in everyday life, you are not used to see spots of light. So we have to make some sense out of it and get it some application to everyday life vision that you experience. And what you see actually when you look at your environment, you see shapes, you see contours, you see edges. That's what you see, right? Now, how can you quantify the information that is in an edge, in a contrasting thing between more dark and less dark, whatever, all right? Or in a photograph, the edges of the photograph. Well, mathematicians have developed what are called Fourier transforms. And by Fourier transforms, they were able to assign so-called spatial frequencies to the edges. How can you design now a stimulus that varies in spatial frequency? You do that with what is called a grating. So the stimulus now, instead of being a spot of light, is a grating that is very dark at some point, then becomes lighter, then completely light, then it gets darker again, and you have cycles, you see? You have cycles of the brightness in the grating. And now you can vary, you can define now a special frequency, because if this spot is, let's say, it represents 1 degree of visual angle, and you have 3 cycles within it, the spatial frequency of that spot, it will be 3 cycles per degree, right? And you can make another one that will be 10 cycles per degree. And you can make even another one that will be 15 cycles per degree, and so forth and so on, all right? You have one here that 29 cycles per degree. So these are stimuli that approach more of what you are seeing in everyday life. You are analyzing contours through this Fourier transform, all right? Now, when you use this stimuli to plot the receptive field of the ganglion cell, you will find that from that standpoint, you have again 2 different other kinds of ganglion cells. You have a ganglion cell that is called of the M type and ganglion cells of the P type. What M and P means, M means magnocellular, which means large cells, and P means parvocellular, which means small cells. We are always dealing with retinal, with the ganglion cells, all right? And these 2 types of cells have very different characteristics. For instance, you have here the size of the cells. I already said that the M cells are large and the P cells are small. Right? Oh, I have it distributed in column. In any way, the conduction velocity of the axons of these cells, the M cells are fast. Where they are located in the retina, mostly in the periphery of the retina. What is the response to a stimulus? It's usually transient, which means they react mostly to the on and to the off of the stimulus. When you present the stimulus and we remove the stimulus. At those spots in time is where they react mostly. All right? Now, what about their spatial frequency? It's only since these neurons only react to very low spatial frequencies, which means that they are not too interested in the spatial frequency. All right? Only very low grade, maybe 2-3 per second, 2-3 degree cycles per degree will the cell react. On the other hand, you can do another trick with these stimuli, which is not only present them fixed in space, but you can move them back and forth with a certain frequency of movement. That means that with these stimuli, not only you can explore the spatial frequency, but vary the spatial frequency, but you can also vary the temporal frequency. And these neurons, the M cells, these ganglion cells, are highly sensitive to temporal frequencies. And finally, what about the reaction to color? The M cells can care less. They don't see color. They're not interested. All right? So keep in mind these characteristics and see the opposite essentially in the P cells, which are small, that the conduction velocity is slow, that they predominate in the central retina, that their response is most sustained, which means that while the stimulus is there, the cell keeps on reacting to it. Remember that the other was transient at the on and the off. How about spatial frequency? Not only they are very sensitive to spatial frequency, but they are specially tuned to particular spatial frequencies. What does it mean? That there are ganglion cells that are particularly sensitive to 10 degrees per second, others to 5 degrees per second, others to 3 degrees per second. They are finely tuned. Now, what about temporal frequencies? They don't care much about it. Low sensitivity. How about color? Oh, well, they see color. These neurons see color. Because if you plot the receptive fields, you will see that this results in what is called the single color opponent cell, in which any stimulus in the center, if it's red, will excite the center, and a stimulus that is green will inhibit the surround. So this is a single color opponent cell. Red excites the center, green inhibits the surround. Now, you already have the idea here that what we were discussing initially of the two types of retina, the central retina and the peripheral retina, follows the two types of ganglion cells, because in the central retina, that where the cones were predominating and they have very low sensitivity by high acuity and they saw color, whatever, there is where the ganglion cells reside also, in that central portion of the retina. So whatever is processed by the various layers of the retina at the central level, they result in a particular receptive field of particular ganglion cells that will be of the P type. Now, have it clear that when we discussed the spots of light, we said the two types of ganglion cells were the center on surround off, or the center off surround on, right? Here you have this other kind of classification into M and P cells, but each one of these categories could be of the center on or of the center off. So the two ways of examining it, they overlap each other. So we just have seen the various types of ganglion cells characterized by different receptive fields, depending essentially on the stimulus that you are using. With spots, you got the center surround antagonistic organization, and with gratings of various frequencies, you have also the characteristics of these cells in terms of their response to temporal and spatial features of the stimuli. Before summarizing this information, I must tell you about some relatively recent findings, not of yesterday, but recent in history. And that's the very interesting fact, that there are some ganglion cells that contain visual pigment. This visual pigment at this level is called melanopsin. Now you are familiar with visual pigments at the receptor level. Now we are talking about the ganglion cell that has pigment and that reacts directly to light, impinging on it. This is an extremely interesting finding because it may serve as the basis for a lot of non-visual functions of the effect of light on the organism. For instance, when we discussed initially the pupillary constriction to light. Well, this is not a visual, doesn't give rise to any visual experience, but this effect of light on the organism and is a very fast reflex. So it's very possible that light bypasses the entire networks of the retina and directly activates ganglion cells that contain this melanopsin. This also could be true of circadian rhythms. Circadian rhythms that are governing many functions of the organism, entrained by the light-dark cycle of the day, the 24-hour plus cycle. So it's possible also that circadian rhythms are activated by light directly on particular ganglion cells that contain the melanopsin. Finally, it is well known the effect of light on mood, for instance. There are some forms of depression that benefit a lot from a light treatment. And when the weather is gloomy or whatever and gray, people get more depressed. All right? These obviously are not visual functions, but there is no question that this effect of light acting on the organism, and it's possible that that kind of function that is not resulting in a perception of something doesn't need all the processing in the retinal network and starts with activation directly of ganglion cells containing this melanopsin pigment. All right, now, I think this ends this first part of the visual system, that you realize that we are talking about the retina, the peripheral portion of the visual system, and only in the next 2 hours we will discuss the central portion of the visual system. So far, for us take-home messages, you pay attention to the special attention, as I said here, pay attention to the effects of lesion on the pupillary light reflex that we discussed initially. Then the lens system that focuses the visual target on the retina, and that you have these defects in myopia and in hyperopia and presbyopia, as discussed before. Now, the concept of visual field for each eye, right? That was maximally extended for movement detection in the 100 degrees temporal, which is out there, and only 60% nasal, and all the other measurements that we discussed initially. I think you should have some idea of that. Then we went into the retina, and we noticed the imperfections of the retina. First, that it's inverted, that the light has to traverse all the layers to reach the receptors, and also the chromatic aberration, and the presence of the blind spot, which is the head of the optic nerve that doesn't contain receptors, and how nature correct for that. For the inversion of the retina, you have the fovea and the foveola. Then for the chromatic aberration, you have the yellow pigment in the macula lutea, and for the central mechanisms to compensate for the blind spot. We said that the pigment epithelium mediates active transport between receptors and the blood, and stores vitamin A, and phagocytes the debris of the shedded discs of the receptors. Then we said that the central part of the retina, which is about 20 degrees, which is the macula, right? around the fixation point, and the peripheral retina are structurally and functionally different, primarily due to differential distribution of the receptors, because the cones predominate in the central retina and mediate photopic or daylight vision, whereas that with the characteristics that you know already, the low sensitivity, high acuity, and color vision, and the rods that predominate in the peripheral retina and mediate scotopic night vision with high sensitivity, low acuity, and not much color perceived. A few more messages. The visual pigment is a protein molecule with an embedded chromophore group, which is the 11th cis retinal, that is bent at the 11th carbon, right? That the visual process, no matter what visual process, starts singly by the action of photons on the chromophore group, straightening the carbon chain and transforming it therefore into an all-trans retinal. From there on, there is a series of second messengers that result in the hydrolysis of cyclic GMP that closes sodium channels and thereby hyperpolarizing the cell, the receptor cell, decreasing the release of its transmitter. Totally counterintuitive. Then you have that the further information is being processed in the neural retina that consists of the bipolar cells and the horizontal and amacrine cells, and eventually results in the characteristic centers around antagonistic receptive field of the ganglion cells that can be of one type and the other type. All right, that may be of the on-center off-surround, or can be just the reverse. That we discussed at length. Now, essentially what that is doing is sampling the level of brightness of the target and the various parts of the stimulus, all right? Now, each of the two types that we discussed just now can be of the magnocellular M cells or parvocellular P cells categories that again send information as separate channels to the brain. Next. Yes. Now the P cells predominate in the central retina, are finely tuned to spatial frequencies, and are single color opponent cells. For example, red excites the center, green inhibits the surround. Therefore, they sample form through the spatial frequency and color through these single color opponent cells. On the other hand, the M cells predominate in the peripheral retina, are highly sensitive to temporal frequencies, and are insensitive to color. Thereby, what are they sampling? They are sampling essentially movement and growth contours. Finally, I didn't mention that, but it's just on the way, and perhaps we'll pick it up next in the next lecture. A retinal lesion. Suppose you have a lesion in a particular piece of retina. It can be caused by retinal detachment or a trauma, whatever. All right? That results in a very restricted piece of blindness in the visual field of one eye, of the affected eye, and that's what is called a scotoma. So the scotoma is a blind spot in the receptive field of one eye that would be characteristic of a retinal lesion. Thank you very much. We will continue